The Southeast Center for Integrated Metabolomics, or CSIM, is a service center that provides targeted and global metabolomics and lipidomics analysis for multiple sample types, from biological samples from human research studies to plant samples for environmental studies. Global metabolomics is an unbiased way of looking at many types of metabolites in a given system and is combined with multivariate statistical analysis to identify biomarkers. It is often called a hypothesis-generating approach. Targeted metabolomics involves measuring and quantifying specific metabolites. In targeted metabolomics, we typically have a hypothesis about a specific metabolite, and we want to know if that hypothesis is true or false by measuring the metabolite expression. Our mission at CSIM is to provide world-class metabolomics and lipidomics services using state-of-the-art technology while providing superior customer service. We work with investigators from start to finish, meaning that we can help design experiments, define how best to preserve the metabolites and lipids, perform all extraction, and conduct initial bioinformatics. It's almost a one-stop shop for all needed services. Our labs are equipped with a combination of high-resolution, high-mass accuracy, ultra-sensitive instruments, which aid in the identification of unknown compounds and detection of targeted compounds. CSIM also provides education opportunities to aid the young metabolomics investigator. These opportunities include summer or winter school in metabolomics, as well as short courses that provide different conferences throughout the year. Metabolomics relies heavily on the use of standards to accurately identify metabolites to the most stringent levels, and we follow all best practices defined by the Metabolomics Society for Identification. We use retention time and mass accuracy curated libraries, which are always expanding, and as more identifications are revealed, this can help elucidate real mechanisms and aid in understanding disease progression. The process of analysis begins with the samples. We work with investigators to obtain all the key metadata so that we can appropriately plan extractions and randomizations. Once we have defined all the project goals, samples are extracted in batches of around 50. We include quality control specimens, blanks, and extraction blanks that are used to assess each batch for quality. In addition, we add several internal reference standards as well as injection standards, which allows us to monitor our extraction efficiency, reproducibility, and instrument performance. All of this ensures the highest quality data is always obtained. After data collection on the instrument, we implement our open source processing pipeline to evaluate all reproducibility metrics first and then move on to identifying metabolites and unknowns across all samples. It is true that for every hour of instrument time, it takes roughly 7 hours of data processing time. Finally, statistical analysis is the last step, and our center performs initial statistical analysis that includes univariate as well as multivariate approaches. In addition, we also can work on more complex approaches such as machine learning and pathway mapping to help our investigators better understand the unique chemical signatures that arise from their data. We look forward to working with you as we drive metabolomics and lipidomics science into the future.